I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. My name is Jesse Lawler. I am your host, and this is the 161st episode and the last episode of the year 2016 of this podcast dedicated to the ongoing betterment of your brain by any and all means at your disposal and at all times and under all circumstances, including rather nasty circumstances like just having suffered a stroke. Hopefully this does not apply to you or any close friends or family members, but stroke is a very, very common condition, probably more so than a lot of people realize. And in this episode, we're going to be speaking with Dr. Thomas Carmichael, who is a neurologist and a leading researcher into stroke and particularly the rehabilitation of the brain after a stroke has happened. Probably needless to say, this is a situation where an ounce of prevention is worth a pound of cure, but these things do happen, and gaining a better understanding of how the brain can repair itself after a traumatic situation like a stroke is obviously useful information in its own right and could potentially open the door to new forms of therapy for stroke and other brain conditions. It's actually not a doom and gloom interview at all, despite the subject matter. There's actually some really interesting findings and also some interesting parallels and contrasts to how the brain behaves when repairing itself versus during development, learning, or other situations where the brain is changing. But that'll be in the main interview. If you hang around until the very end of the episode, I'm going to tell you something that deals with the brain and sneezes. This is a weird one, so you'd be the first on your block to know this new sneeze trivia that'll be at the end in the ruthless listener retention gimmick. But first, as usual, let's kick things off with This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
So one thing I am hoping to get to do later on this year is to go into a quiet room. If I do a Smart Drug Smarts episode will certainly result from this. Still in the works, but there are in the world some very, very quiet rooms used for testing acoustical equipment, things like that are so quiet, so free of any echoes that actually it is supposed to be psychologically weird and oftentimes kind of disturbing to even spend any time inside of them. But anyway, as somebody with a podcast and an interest in audio, I oftentimes have my own ears prick up when scientists are studying sound. And just a couple of weeks ago at the 172nd meeting of the Acoustical Society of America and the 5th joint meeting with the Acoustical Society of Japan, there was research presented by Takahiro Tamasui, an associate professor from Yamaguchi University in Japan, looking at various noises and how they affect cognition. We've had a couple episodes previously about white noise and brain entrainment, different ways of potentially using sound to enhance concentration. It's also no surprise that sound can be a big detriment to concentration if it's the wrong kind of sound. Babies crying, fingers on chalkboards, things like that are famously distracting and unpleasant to hear, but the level of subjective annoyingness has rarely been scientifically studied. Yamaguchi and his research team conducted experiments using what's called the oddball paradigm, where a stimulus that didn't fit, or multiple stimuli that didn't fit, had to be picked out from a much larger group of stimuli. This could be done with visual cues, with auditory cues, even with smells and other stuff, I guess, but for these purposes they were auditory, picking out things like a 2,000 hertz tone among a series of 1,000 hertz tones, and at the end of the trial, the subjects also rated their personal level of annoyingness. They ranked each of those sounds on a 7-point scale. But while the research subjects were getting these stimuli, these things that they were supposed to be paying attention to, they were also having audio played in their earphones. In some cases, it was male and female voices speaking. In other cases, it was white noise that was kind of the same frequency ranges as if people were speaking. They did a series of experiments of this nature, and interesting but possibly not surprising, the more meaningful noises were a lot more distracting, made the people perform worse on the oddball test. So hearing somebody speaking in a language that you can comprehend, even though you might not find that to be the most annoying sound in the world versus like a woodpecker tapping on your windowsill or something like that, it's actually easier to filter out the meaningless sound of a woodpecker tapping than it is a conversation going on that you can actually hear and understand, said Tamasui. When designing sound environments and spaces used for cognitive tasks, it's appropriate to consider not only the sound level, but also the meaningfulness of the noise that is likely to be present. Because it is difficult to soundproof an open office, a way to mask meaningful speech with some other sound would be of great benefit for achieving a comfortable sound environment. As a quick personal note, this is why I very, very much approve of and endorse high-tempo instrumental music when I'm trying to get work done versus anything that has any lyrics in it whatsoever.
Smart Drug Smarts, the podcast so smart, we have smart in our title, twice.
Going into the international iTunes grab bag, grab some 5-star reviews from Israel and Australia, respectively. Mosh Becker says, "Fantastic podcast. You just make me think better and clearer. Really appreciate it." And Eartho says, My biggest fear is my brain idling away in Sydney's malicious traffic with nothing adequate to do. Fortunately, at least for a while, I have Smart Drug Smarts, the best podcast I have to justify my day. Well, a big thanks to both of you. Thanks to everybody that's been leaving reviews, spreading the word, laying down the occasional tasteful graffiti. I just fired off a Brain Breakfast newsletter earlier today. If you're on the newsletter list, you've already seen that. If you haven't yet, but you want a good way of keeping up on what's going on in Smart Drug Smarts, or in this case, hearing both my thoughts on 2016 and some anticipations for 2017, you can sign up for that at smartdrugsmarts.com/newsletter. It's hard not to be a bit wistful at year's end, and this has been a particularly good year for Smart Drug Smarts. I can't help but remember about this time last year when we were really still kind of struggling episode to episode to make sure that we had a good interview ready for the next week. That is a problem that we've largely solved in 2016. It's nice to be entering the year with a big stack of interviews ready to go, and also a whole lot of interesting ideas, many of which our listeners applied for future episodes that we're interested in doing. So lots of good things upcoming. I mentioned some of the upcoming interviews in last week's episode 160 if you didn't catch that. Looking forward to summer 2017. We'll be having the 2-year-old birthday for Axon Labs, the retail arm of Smart Drug Smarts. And we actually had the podcast's 4-year-old birthday. It went unremarked upon in November. I should have said something at the time, but it kind of snuck by without my notice. But yeah, we're actually 4 years old now. So thank you, everybody that's been a long-time listener or a short-time listener that just found us. Appreciate you all. All are welcome here, of course. And if you don't know what I'm talking about with Axon Labs, that definitely marks you out as a newcomer because it's something we've been talking about for a while. But we've got a couple of supplemental stacks over at axonlabs.io, general metabolic and mitochondrial health in our stack Mitogen, and a cognitive specific stack built around aniracetam called Nexus. A lot of people on my gift list got that in their Christmas stocking this year, so looking forward to another year of witty and pithy conversations, keeping everybody on their cognitive toes. But that's it for now, so let's jump ahead for the very last interview of 2016, coming up next.
Smart Drug Smarts.
So I'm about to be speaking with Dr. Thomas Carmichael of the Carmichael Laboratory at the David Geffen School of Medicine at UCLA. Dr. Carmichael is both a neurologist and a neuroscientist. That's a distinction that we probably don't talk about enough. It's one of those ones worth bringing back up. A neurologist is somebody that has an MD and is actually like a doctor specializing in areas of the brain and nervous system. A neuroscientist is more of a research academic focused, probably has a PhD, but need not be a medical doctor. But Dr. Carmichael, who we're about to speak with, is both. And his particular focus of study is on neurorehabilitation after stroke. He looks at both the molecular and cellular mechanisms of neural repair, and he came across our radar screen as we were reading on some of his work studying axonal sprouting. Axons, as you probably know, are the long outbound messaging primary offshoot of the body of a neuron. You can kind of think of them as the really tall cell towers from which the neuron makes its broadcasts. Anyway, really interesting conversation about to happen. You can kind of think of this as a sibling episode to the episode on traumatic brain injury we had a month or so ago. A stroke would be a non-traumatic brain injury. It's definitely not something you want to have happening to your brain, but it's minus the percussive force, the 2x4 to the forehead or whatever it is, that puts the trauma in traumatic brain injury. And as you'll hear, there are both some important similarities and differences between the brain's responses to those two types of injury. So with that as preamble, let's jump in with Dr. Thomas Carmichael.
As I trained as a neurologist, I recognized that really in the entire field of neurological therapeutics, there are no drugs or approved therapies that promote brain repair. Whether that's in stroke, multiple sclerosis, Parkinson's disease, epilepsy, there's really nothing out there that exhibits a biological underpinning of brain repair and tissue recovery. I really realized that the science underneath tissue regeneration, the CNS, is really fascinating. It involves basic biological principles of neurodevelopment, but also aging and how those two interact to influence the molecular response of the aged or middle-aged brain to repair and recovery.
It sounds like that's implying some age differences. If there was a brain injury in a younger person, how would that affect the brain's response?
At the extremes, we know that there's a substantial difference in how the brain responds to injury. So, for example, many of your listeners will recognize anecdotes of kids under age 2 who can have substantial brain injuries and exhibit almost a complete recovery. There are fascinating examples of children under age 2 who've needed a hemispherectomy for intractable seizures, one whole half of the brain gone, and they make honors English in 8th grade. It's really remarkable. The next cutoff is approximately age 6, where between 2 and 6 you can still have a really fabulous outcome with a substantial brain injury or a substantial resection, but it's not complete and there are noticeable deficits. And then from 6 to somewhere in late teens, there's still a fairly robust recovery, and the brain recovery then plateaus for most of late young adulthood and middle age, and then declines again in aging. And so there are these sort of time epochs in which there is substantial plasticity and recovery, and then a graded series of decreases.
Do we know what tips somebody over the edge into that final stage of the brain's ability to self-rehabilitate, where should they get an injury, the brain will probably never recover to really what it once was?
Well, we know that the natural course of that change is hardwired at the molecular level. So when you look at regenerating brain tissue, for example, neurons that might form a new connection after stroke, and you compare their molecular expression profile, basically the genes that they activate or turn off, between an aged brain and a young adult brain, you see substantial changes just in the natural state. So the aged brain, its molecular mechanisms to respond to injury and repair are substantially and surprisingly different than even the young adult brain. So whatever the change is, it's fundamental, and it occurs both at the phenomenological level, where we just see these people, or in the lab, these experimental animals recover less well with age, but it's also very much present at the molecular level.
You and your colleagues have made a lot of discoveries about a chemical called GDF10 that naturally occurs within the brain. Tell us about what you found there.
With those discoveries, we were particularly interested in the aged brain and what molecular systems it might activate that perhaps signal a limited recovery seen in that state. And so we did a series of experiments over the past 10 years that have all been published, in which we tried to identify what we might call the regeneration transcriptome. What is the gene expression profile of a neuron that's stimulated to form a new connection after stroke? And then we compared that transcriptome between the young adult and the aged with the specific forethought that we would really go after those genes that were really upregulated in the aged brain. And one of them was GDF10, or growth differentiation factor 10. And it was also intriguing to us because it really had no known role in the adult brain. And we thought, here is a system that possibly is unique to both aging and a regenerative response, and we may have kind of an avenue of pursuit there because it isn't known, and it seems to be at a crossroads in our molecular studies of a condition that might lead to repair if adequately stimulated.
The fact that this is GDF10 and not GDF1 or just GDF implies that there's at least 9 other sibling compounds. What do we know about the related compounds in this family and what they're doing?
And it's a divergent family member of this GDF family. And then the GDF family itself is part of the TGF-beta, or transforming growth factor beta superfamily. So that's a large family of cytokines that include activins, GDFs, TGF-beta, and other molecules that have a role in practically every aspect of the body's tissue response to development, aging, and stress. The GDFs have been described mostly in bone development and bone function. That's where their role is most well known. They haven't really had a well-established role in aged brain repair. There's a factor GDF11, which made transient prominence in a series of articles that suggested it was the aging factor. And your listeners may know that. A fairly high-profile series of papers in Nature Medicine and Science, some of which are somewhat clouded because they've been hard to replicate, but it does indicate that the GDFs have a role in aging and possibly aging tissue repair.
Is there a normal background level of GDF10 that's around in an uninjured brain, or is it something that really only shows itself after an injury?
We could detect it at the transcript level, meaning that the gene was turned on at a very low level in the normal brain, but it was profoundly upregulated, in our case in stroke, in the tissue adjacent to the stroke site, and that's the site which undergoes most of the repair and recovery in experimental models of stroke and in humans. So it is there. Its normal mechanism of action, whatever it's up to, is not really well known, but it's really profoundly induced in stroke.
How does the brain or the brain cell know when a stroke has occurred? Like, what is the actual signal of injury at that microscopic level?
There are probably several. So for the field, most of the focus is not on the stroke itself, which is a core of dead tissue, and that's primarily because in humans in particular, the core of that stroke, ground zero of low blood flow when a clot gets lodged into the brain, is really past any therapeutic intervention within an hour or so. And the focus really is on the tissue that borders the stroke. And in the repair and recovery field, it's this peri-infarct tissue that is where GDF10 is induced, where there is the process of forming new neurons, where angiogenesis is prominent. This is where tissue regeneration really occurs. occurs.
Let me make you back up there. Peri-infarct, that's the word for sort of the adjacent tissues? That is a term I'm not familiar with.
Sure, it's mostly just a spatial term, peri meaning around,
Okay.
and infarct meaning the stroke. So it actually carries with it no intrinsic meaning.
Gotcha.
It's simply a descriptor. So the signals from the core, which we can't really do much about, this area where everything dies in a stroke, the signals from that to the tissue that survives. And we know there are a couple of cellular signals that are transmitted there. One is an intense set of inflammatory signals. Secondary inflammation is activated quite strongly in stroke, and those inflammatory signals, both white blood cells and cytokines, diffuse out into this peri-infarct tissue, the surrounding and surviving tissue. A second series of signals are more on a molecular level. In human stroke, there is a clot and an artery, and the tissue that was totally dependent on that artery for its oxygen dies. But there are zones around it in which only part of the blood flow came from that artery and part came from neighboring arteries, or so-called collaterals. So that tissue survives, but it took a hit. It did sense a little bit of low blood flow. And when it gets its blood flow back, there is what's called a reperfusion event. So a tissue zone that was originally a little low in its oxygen then experiences a reflow. And that leads to a wave of reactive oxygen signaling that is in itself a molecular event to signal regeneration, that the free radicals and other molecules generated during tissue reperfusion are themselves some of the early signals for tissue repair.
Would this same sort of response also apply to a non-stroke injury? So correct me if I'm wrong here, but like traumatic brain injury is when you actually take a whack to the head. Something like a stroke would be still considered a brain injury, but of the non-traumatic kind. Would you see something like GDF-10 deployed in the case of more traumatic injuries?
That's a good question. We have not looked. That's on the list of things to do. There are many events that don't transfer from traumatic brain injury to stroke or the other way, and one of the reasons for that is some of these signaling events that I mentioned are unique to stroke. This low flow state and an adjacent area of reperfusion injury appear to be particularly important for events that involve angiogenesis and neurogenesis, or the formation of new blood vessels and new neurons. And those events are not as robust in traumatic brain injury as in stroke, and it may be because the mechanism of injury is different.
One thing that I saw that was both counterintuitive and really pretty deeply interesting was that we think axonal sprouting, that sounds very similar to what goes on in a learning process, kind of laying down new neural pathways. But apparently what's going on in learning versus injury recovery is really quite different. Can you talk a little about that and how that was teased apart?
What we do know is that the molecular mechanisms that support neurodevelopment, or the formation of the brain and its connections, are not simply recapitulated in the formation of new connections after stroke. There is a kind of a buzz term in the field that regeneration recapitulates development. And what my work and others has shown is that's not the case. There is overlap, but regeneration is really a unique cellular and molecular event that has some elements of the original developmental program in it, but also has unique elements that reflect the fact that it's occurring after injury and in an adult. So it's not simply that recovery in stroke or in head injury is a reactivation of a youthful brain, if you will. Mm-hmm. It is instead a unique regenerative event. But to directly answer your question, there are some very interesting parallels, not with development, but with learning and memory processes in recovery. And so my work and others have shown that some of the synaptic signaling events that enhance learning and memory also enhance recovery after stroke. And so we had a paper in Nature in 2010 where we looked at one system that involves extrasynaptic or tonic GABA signaling. And that's an avenue to promote recovery in stroke, is to manipulate tonic GABA signaling. And another one is to use positive allosteric modulators at the glutamate receptor signaling system, the so-called AMPA receptors. And both of those originally hit the radar screen based on their role in memory formation, and they do have a role in stroke recovery.
Are there any downsides to the upregulation of GDF-10? If somebody had too much of this present after an injury, or present at all, I guess, in a non-injury state, are there going to be negative consequences?
Yes, there really will be. This will be one of the fascinating areas of this field in CNS regeneration as it evolves. There are a couple of potential downsides. They're all potential. We don't really know since we didn't directly test them. But as a TGF-beta family member, GDF10 will have an effect on many tissues. And so systemic administration of such a powerful and pleiotropic cytokine is likely to have off-CNS effects. And so if this were to be translated to a small molecule agonist, say, we showed that it was the TGF-beta receptor 1-2 complex that was necessary. If a small molecule agonist to that complex were generated, it would have to be of short enough duration, if given systemically, that it didn't provoke these profound systemic side effects, or alternatively locally delivered to the site in the brain where its activity is necessary.
How long does GDF10 remain in circulation in the body in the aftermath of an injury? Is it something that there's like a quick acute spike when the injury occurs and then it fades off, or does it keep being secreted as the injury is still present?
What we found in stroke in the rodent is that it's induced at day 7, so it's turned on, and then it's turned off again at day 21. Then we looked in non-human primates and we looked in humans, and it may be a longer time scale there. So we found months after the stroke in primates that it was still up, and also in humans. So it may be that the time scale differs between rodent and primate.
That is a much longer time than I expected you to say as far as when GDF10 first shows up on the scene on day 7 following the injury. What's going on there? Why the delay?
Well, it could be that it's induced early and the signal is lost because there's so much damage and so many different kinds of processes going on in the first 3 to 5 days after stroke. Often we don't even look then because the inflammatory signals and the signals of secondary cell death are so strong that it's hard to resolve the initial signals of recovery and repair. So we kind of spot the brain a week
right.
after a stroke, because then you clearly know that death and destruction are over by day 7. And when you start to study something that's induced, it's more likely to be implicated in repair and recovery.
What other raw materials does the body need for axonal growth? The signal is the GDF10, but what is the body going to need as far as dietary constituents, things that are delivered on the bloodstream, stuff like that?
There's probably not much we can influence on a dietary level, because this is, all told, a relatively minor response in the brain in terms of the hundreds of thousands of miles of axons and connections, the millions of neurons that we have. And instead, the axonal sprouting is just and formation of new connections is a small part of that. But one thing we do know is the currency of the realm, of course, in the brain is neuronal activity, neuronal signaling. And so the one thing that people can do that dramatically influences recovery is to be very active in using the circuits that are damaged. So when you look at even physical medicine, neurorehabilitation, the more active people are in using their impaired limbs, the better they recover. And as we start to this sort of new world of repair therapeutics that might unlock the ability to form new connections, there's going to be an exquisite interaction between a drug that stimulates the brain to form new connections and then the activity patterns of the system in the brain that is exhibiting the sprouting. So that if you were to engage axonal sprouting in a patient with a drug and then sit them on the couch and have them watch The Big Bang Theory for a couple of weeks, you would probably find that the connections were aberrant.
Right.
Whereas instead, if you gave the connections specific instructions by a rehabilitation paradigm in the injured limb or the impaired limb, you're likely to instruct those connections to form in the appropriate circuits. So there's a growing awareness that we're going to probably have to couple some of our repair therapies with sort of instructive activity.
In the papers that I read about your work, it was talking specifically about the growth or the lengthening of axons. But of course, for every axon, there's hundreds or thousands of dendrites also on a neuron cell. Does GDF10 do anything for dendritic growth? Is there a parallel there?
You know, that's a great question. We didn't study it. The best way to study that involves in vivo optogenetic imaging of dendrites. I think that's the most convincing way to demonstrate that. And so we have that technology going in the lab, and others have also been pushing that. And that end of the field is still at the descriptive medicine end. What happens to dendrites over time in stroke naturally? We're sort of just finishing that descriptive stage. So now we can start to characterize what these molecules might do to enhance or alter dendritic changes after stroke.
What studies would you like to see conducted or to conduct yourself next? What are the big remaining question marks in your mind?
There's a couple of big ticket items in this field. And one is, can you deliver therapies like we discovered here in chronic stroke? So the prevalence of stroke in the U.S. is 7.1 million people.
Wow.
For comparison, comparison, Alzheimer's is only 5. And so for all the hullabaloo we hear about Alzheimer's and its prevalence, there are more people living with stroke. And if we could deliver a therapy to chronic stroke, we would really impact the lives of a lot of sufferers. So that would be one step, is can we model chronic stroke in the rodent and test therapies that might improve chronic stroke in humans. A second is to get down what you indicated before, and that's delivery and dosing. Outside of GDF-10, there are many other approaches in the field to enhance recovery, and many of them will trigger other events. Many of these approaches are sort of loosely termed pro-growth. They stimulate brain growth. And the best example of a pro-growth condition is cancer, and many of the same molecular systems are actually active in oncogenesis and in metastasis. And so it will be an important next stage to understand how we can deliver what we might term pro-growth brain therapies and not be generally pro-growth to the rest of the body.
Smart Drug Smarts.
And a very big thank you to Dr. Thomas Carmichael for taking the time for that conversation. One of my favorite things about that conversation actually was the number of things that we didn't have answers to yet just because the experiments have not yet been done. One of the constant drumbeats in neuroscience is the complexity of the brain and the number of things that we still don't know, and how in some ways it's maybe still the greatest unknown frontier in scientific study. So in some ways it's good news when there are these things that we could easily look for the answers to that nobody's had the time or the inclination or maybe the funding to look for yet, because in some ways these are still easy wins to be had, rather than things waiting for some massive epiphany or massaging a little probabilistic finding out of a massive wealth of data. It's just kind of nice that sometimes the answer is, well, we don't know because we honestly haven't had time to look yet. But now, from strokes to yawns, let's jump ahead to the ruthless listener retention gimmick. And by the way, I apologize. At the beginning of the episode, I said that this ruthless listener retention gimmick was going to be about sneezes. So if you're a big sneeze fan, and I'm kind of pulling the rug out from under you because now this is going to be about yawns. Yawns sound a lot more boring, but this is actually interesting. So hopefully you'll forgive me. This was not an intentional bait and switch. I was thinking yawn. I said sneeze. I don't know what happened.
Smart Drug Smarts. Ruthless listener retention gimmick.
And I quote, characterized by a powerful gaping of the jaw with inspiration, a brief period of peak muscle contraction, and a passive closure of the jaw with shorter expiration. Such is the definition of a yawn. But it has been said of yawns that yawning may have the dubious distinction of being the least understood common human behavior. And despite the fact that people have been yawning forever, and it's done in all cultures, and in fact it's done well beyond the human species, even reptiles, even fish seem to yawn, although it's not necessarily agreed that it serves the same purpose in reptiles and fish that it seems to in mammals and birds. But yawning is pretty darn ubiquitous, and yet it's not always agreed upon what yawning is actually for. It must be important because everybody's doing it, and it has been evolutionarily conserved. It hasn't been bred out of the population or thrown by the evolutionary wayside. It's pretty well accepted that to a certain degree, yawning is contagious, quote unquote contagious, within humans. If you see somebody else yawning, you're a lot more likely to yawn yourself. And that's led people to speculate that there might be some sort of social signaling going on, at least within our species. But other research has suggested that yawning might be an adaptation to enhance intracranial circulation and brain cooling. In other words, if your brain is getting too hot, yawning is your body's way of turning on the AC. The neural structures that drive yawning, that actually make yawning happen, appear to be located within the brain stem, the oldest portion of the brain. But the authors of this paper speculated that the neurophysiological consequences of yawning might affect the brain much more globally. So it's an action by the brainstem, but for the benefit of the brain generally. Trying to get a handle on what exactly is going on with yawning, these researchers took advantage of the fact that yawning happens so widely across the animal kingdom, and instead of shrugging their shoulders and saying, they decided to look at the variance in the way that different animals yawn, and they chose to look at yawn duration as something that would be a common and measurable trait in all yawns, where they might find some interspecies variance and also variance among members of the same species. And this was kind of cool because they actually did their research simply by going to YouTube from the sounds of it and using YouTube as a search engine for a variety of animals yawning. They found many, many different YouTube videos of yawns. They threw out anything where they didn't have both the beginning and the end of the yawn, and they wound up with 205 full confirmed yawns from 177 individual animals across 24 taxa, including African elephants, camels, cats, chimpanzees, gibbons, hedgehogs, marmosets, rabbits, rhesus monkeys, sheep, squirrels, walruses, and, of course, humans. There was a lot of publicly available data on different animals' average brain weight, brain volumes, approximate number of neurons in different regions of the brain, that sort of stuff. So they were able to do a statistical analysis of these results. And what they found was that the average duration of a yawn was a very robust predictor of both a mammal's brain weight and the number of cortical neurons. Consistent with these findings, primates tended to have longer and more variable yawn durations than those of other mammals. Neither the animal's body size nor anatomical structures like the size of the skull or the jaw seem to be driving these effects, because gorillas, camels, horses, African elephants, all things have bigger heads and jaws, and in some cases physically larger brains than humans, but all had shorter average yawn duration. And again, it was the number of cortical neurons that was the most significant factor. So some compelling evidence for yawning as a brain temperature control, although the jury is still out. And I'll tell you, while reading this study, not being particularly tired, I caught myself yawning a couple times. Even just thinking about yawning this much makes a person want to yawn. But next time your professor catches you yawning in class, just say, hey, I've got a big brain and I don't want it overheating.
We like to think of ourselves as a digital speakeasy for brain hackers, but you can call us Smart Drug Smarts.
Okay, you made it all the way to the end. That is episode number 161, now complete. Thank you for hanging around with us. If you enjoyed this episode and are looking for the links or any of that relevant stuff, you can find that all online at smartdrugsmarts.com/161. Last week, if you missed that, we talked with Dr. Nicole Avina in episode number 160. And next Friday, our kickoff episode for 2017, I can't tell you exactly what the episode topic is going to be because I haven't decided yet. We got some good options, but it'll be brain relevant and hopefully brain applicable to your brain in particular. More specificity than that is anyone's guess, but join us back here next Friday, same time, same podcast. New year, but with that same unflagging commitment to helping you fine-tune the performance of your own brain. Have a great week, conserve brain cells on New Year's Eve, and above all things, stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.